Hello everyone. This session will cover or enable you to get an overview of mitochondrial structure and its functions. And it is one of the organelles that is responsible for energy conversion. Like the other organelle that is also involved in energy conversion is chloroplast, which is solely found in the plants. So let us look at the learning outcomes of the session. Mitochondria with a double membrane. So this is another organelle which has a double membrane, like nucleus has a double membrane. Similarly, mitochondria also has a double membrane structure. They are dynamic and plastic organelles. When one says plastic organelles, one can get from one several, and with the ability to move around in a cell, taking help of microtubules. So it need not necessarily be fixed at a particular region in the cell, but it can move around depending on where its function or where it is needed. Mitochondria is able to produce energy by oxidative phosphorylation carried out by the electron transport system and ATP synthase. This is the reason that mitochondria is also called as the powerhouse of the cell. The mitochondria is highly active in metabolism with several metabolic pathways and cycles like TCA cycle, beta oxidation, fatty acid synthesis, etc. All of these taking place in the mitochondria with the help of the inner membrane. Mitochondria has its own DNA. It can undergo biogenesis. Um, it can show what is called as features such as fusion and fission, and it can also associate with other organelles. So therefore now let us look at the various aspects with regard to the mitochondria. So as mentioned earlier, mitochondria basically can be present in several different forms. The most common way by which one generally draws a mitochondria is that it is bean shaped, okay, and it has a membrane, double membrane, and the inner membrane is convoluted. But what is interesting to note is that depending on where the mitochondria is present, you can have the mitochondria, for example, you can see in the cardiac muscle, it is associating itself with the cytoskeleton to carry out muscle contraction, because muscle contraction needs energy, and therefore there are a large number of mitochondria present, and you can see within this itself that the morphology of the mitochondria varies. You have different morphologies within the same uh, tissue type. Then again, what you see over here is a sperm tail, and on the sperm tail, you can see how the mitochondria is wrapped around. So here you can nowhere find a bean-shaped mitochondria. Instead, it is more a tubular kind of shape. Then it is also found to be associated with several different organelles. So, for example, you can see over here how the mitochondria can be associated with the endoplasmic reticulum. And this endoplasmic reticulum, in fact, is able to enable fission of the mitochondria by wrapping around at a particular position on the mitochondria. So, you can see, and also the mitochondria and endoplasmic reticulum interaction is needed for membrane synthesis. That means they give rise to several different phospholipids. So a set of phospholipids is prepared in the mitochondria, a set of phospholipids is prepared in the endoplasmic reticulum, and then from one to the other, the translocation of these phospholipids happen, and then through endoplasmic reticulum, you have the biogenesis of new membranes. Um, Mitochondria is associated also with a very amazing feature, and that is amazing function or phenomenon, which is called as apoptosis. Apoptosis, also called as programmed cell death. That means when the cell, uh, due to some trigger or due to some signal, requires to die, then the process that is followed is the apoptosis, and mitochondria has a major role to play in apoptosis. As you can also see, for example, mitochondria need not be present only at one part. So you can see how mitochondria from the cell body of the neuron is being transported to the axonal end Why you need the mitochondria at the axonal end? Because it's for exocytosis of the vesicles containing the neurotransmitters, energy is needed, and hence for that exocytosis or transport of molecules inside, the ATP that is required at the axonal end is provided by mitochondria. So there is a need for mitochondria to be present at the axonal end. And so using the microtubules and the molecular motors, mitochondria can be transported from one end of the neuron to another end. So this is just an example to basically tell you that mitochondria need not be restricted to only a particular region in the cell. It can be transported from one region to another in region depending on the energy requirement by the cell or by or depending on the function that the cell is carrying out. Now let us go to understanding the structure of the mitochondria. So you can very well see from this electron microscope structure that you have two membranes. The outer membrane, as you can see, okay, and you have the inner membrane. This comprises the inner membrane. Now, interestingly, the inner membrane is a highly folded structure. As you can see over here, it is a highly folded structure, and these folds, enfoldings of the inner membrane leads to formation of what is called as cristae. And within the inner membrane is present the matrix. So all of this inside is what is called as the matrix, while these are the inner membranes that have enfolded to form what is called as the cristae. Now, the region or the inner membrane that is parallel to the outer membrane, okay, is what is called as the inner boundary membrane. So those parts of the inner membrane which are parallel to the outer membrane are called as the inner boundary membrane, while those part of the inner membrane that forms the tubule or a cristae, okay, they are the ones that are called as cristae, and in between the inner boundary membrane and the cristae you can have what is called as the cristae junctions. So therefore, the from these electron microscope pictures, it is very clear that you have a distinct outer membrane and you have a distinct inner membrane. Also, what has been noted from various studies is that the outer membrane composition is very different from the inner membrane composition. The outer membrane has a large number of porins, and therefore the outer membrane is enabling or it is allowing several ions and small molecules of about 5,000 daltons to enter through the outer membrane from the cytoplasm, which means that the space that is there between the inner membrane and the outer membrane, also called as the intermembrane space, okay, has a pH that is very similar to the pH of the cytoplasm, for the simple reason that you have ions and small molecules present in the cytoplasm entering the intermembrane space, and therefore the intermembrane space has a composition very similar to the cytoplasm, and hence the pH also is identical to what is present in the cytoplasm. But the inner membrane per se is very different from the outer membrane. The inner membrane is not permeable to ions and small molecules. So its composition is such that it does not allow or it is much more impermeable than the outer membrane. So that is something that is very interesting with respect to the structure. That means the composition of the inner membrane vis-a-vis the composition of the outer membrane is very different, and hence the functions of the inner and the outer membrane are accordingly different. Now, when you look at the membrane per se, you can see that this is the outer membrane and it is not folded, but the inner membrane, as you can see over here, is highly folded, and these folds are what make the inner membrane have a large surface area. So that is something that is very important for mitochondria in terms of the fact that the mitochondria is the powerhouse of the cell. Now let us understand why it is important that the inner membrane is folded. Okay, So as you can see over here, those portions of the inner membrane that is parallel to the outer membrane, okay, these are the portions where you have proteins or transporters that align with the transporter present on the outer membrane to allow proteins to be imported. So there are several transporter proteins. Apart from the porins, there are very specific translocators that allow molecules from outside to enter, and especially different types of proteins can need to enter the mitochondria along with several other metabolites. So here what you see is the transport across the membranes has to be one through the outer membrane and then through the inner membrane in order to reach the matrix. So if a molecule has to reach the matrix, it has to go through the outer and the inner membrane. So interestingly, what is found is that uh, parts of the intermembrane boundary are actually associated with the outer membrane at those parts. So the cristae becomes defined. You see, if this is interconnected with this, then this is always going to be folded. You see, this portion is not attached to this, but this portion is attached. So it is defining the formation of the cristae, number one. Second, what happens is because you have the uh, um, transport transporter of the outer membrane. So this is the TOM complex, which is also called as the TOM complex. So this is aligned with the inner membrane transporter to basically allow the protein to enter from the cytoplasm into the matrix. So this alignment of the TOM and the TIM help in the transportation, and it is now very clearly defined or compartmentalized. That means this is where this function is going to happen because of the alignment of these complexes. Similarly, you have voltage defined anion channels which are associated again with anion transporters in the inner boundary membrane. So again what you find is that it is at this position that the inner membrane is associated with the outer membrane. Now, these cristae or these tubules that are formed, okay, these are the tubules that generally have the components that make up the electron transport chain. And we all know that the electron transport chain is responsible for the synthesis of ATP. So again, what is interesting to note is this, that the the protein components of the electron transport system are again compartmentalized in a very specific region of the inner membrane. So it is defined where the proteins are going to be, and this is because of the way the proteins are arranged within the inner membrane. It is only cytochrome C that is not associated with the membrane. It is present in the intermembrane space, and cytochrome C also is a part of the electron transport system. So you can see that transport across the membranes is generally through transport complexes present in the inner membrane boundary. We do understand the fact that the inner membrane is impermeable to most ions and small molecules. But if it has to allow specific ions to come in or specific molecules to come in, then it is only through these transporters. Now, one would ask the question, how is it that the inner membrane is so impermeable? What makes it impermeable? There are two main reasons for that. One, when you look at the composition of the inner membrane, you will find that you have a very specialized phospholipid called cardiolipin present. So cardiolipin is a structure where you have one phospholipid attached at the head region to another phospholipid. So because it is attached, you can see that it becomes a more compact structure and it doesn't allow, or the permeability decreases. So depending on the percentage of cardiolipin present in the membrane, you would have more of impermeability. Second, the inner membrane is very special in the fact that this is a membrane that only has about 25% of lipids, but the 75% that is there in the composition of the lipids is the proteins. So again, the presence of the proteins are responsible only for entry of very specific molecules or very specific ions or proteins. So therefore, they would allow substances only when on trigger or only when it has two. Otherwise, it will not. And therefore, the inner membrane becomes very impermeable to ions and small molecules. So effectively, what we therefore can understand is that you cannot have protons just flowing out or in of the inner membrane. You cannot have calcium just flowing in and out of the membrane. So that is not happening. So ions do not go on in and out. Now the cristae junctions. So every cristae you have, just at this point you have what is called as a cristae junction, and these cristae junctions can kind of form a barrier that will localize specific proteins to specific regions of the inner membrane. So therefore, that is how you can very clearly see that how there is a difference in the inner membrane and outer membrane composition, and that structured change is enabling it to differ in its function as well. Now we go to understanding why mitochondria is called as the powerhouse of the cell. So we very well know that within the mitochondria in the matrix, you have what is called as the citric acid cycle or the TCA cycle functioning. And this TCA cycle gives rise to reducing equivalents such as NADH and FADH2. And these reducing equivalents in turn will give its reducing equivalents, that is the protons and the electrons or the hydrides of the electrons, to the electron transport chain for oxidative phosphorylation. So when you look at the mitochondria, you can very well see how from the cytoplasm, fatty acids and pyruvate can be transported through transporters, and those are then within the mitochondria catabolized to give rise to acetyl CoA. So fatty acids are through beta oxidation catalyzed such that you get acetyl CoA, and pyruvate through pyruvate dehydrogenase complex is able to also give rise to acetyl CoA. Acetyl CoA can enter the TCA cycle or what is called as a citric acid cycle, and this cycle has several reactions in which NADH and FADH2 are formed as byproducts. Now these NADH and FADH2 would in turn give its electrons and the protons to the electron transport chain or the electron transport system. So this electron transport system is now going to transfer the electrons that it has obtained from the NADH or FADH. It will transfer it to the next component, that will transfer it to the next component, which will transfer it finally to oxygen. And when it is transported to oxygen, oxygen is converted to water. So basically, respiration, when we understand, this is what is happening. Aerobic respiration is when oxygen is being used as an acceptor, universal electron acceptor, and getting converted into water. Now, in the process that the electrons are transported from one to the other, so you can very well see there are actually four complexes. So you have the first complex, this is complex 1, which is called as the NADH dehydrogenase. This has a redox potential that is able to collect or that is able to accept electrons from NADH. It cannot accept electrons from FADH2. Okay. Now this complex 1 also has a proton pump, and as it accepts electrons and transfers it, it also throws out a proton from the matrix to the intermembrane space. Now we all know that the inner membrane is impermeable to ions. So this H plus will not be able to get into the matrix on its own because the inner membrane is not permeable. So therefore what is going to happen is those protons that are pumped out as electrons are transferred, the protons remain in the intermembrane space. Okay, this is something that is going to help in what is going to eventually lead to ATP synthesis. So you can see that The electrons that are obtained from the NADH, once taken up by the NADH dehydrogenase, is transferred to complex 2, which is succinate dehydrogenase. Oh, uh, uh, sorry, it transferred to complex 3 through a molecule that is called as ubiquinone. Ubiquinone is also a redox molecule. That means it can accept electrons and give electrons. So it is basically taking up electrons from the NADH dehydrogenase complex, and it will give it to the complex 3, which is the cytochrome C reductase complex. The complex 2 is succinate dehydrogenase, which is also part of the citric acid cycle. And succinate dehydrogenase forms FADH2. So FADH2 is able to give its electrons to the complex 3 through the ubiquinone. So therefore, it is missing one proton pump. Now, when electrons from this goes to the second complex is also able to push out protons or pump out protons. When you have electrons transferred to cytochrome C gives it to the complex 4, which is the cytochrome C oxidase. And this cytochrome C oxidase is also a proton pump. So you can see how proton is getting pumped out. Okay. And you can see that all the three complexes are pumping out protons. So therefore, what is going to happen is that in the intermembrane space, protons are going to get collected simply because these protons, once pumped out from the matrix, cannot return back through the inner membrane, and hence they get collected, and this collection of protons is leading to formation of what is called as a proton gradient, and that leads to formation of what is called as a proton motive force. Now this proton motive force that is formed is able to, so you can see this is the accumulation of protons leading to what is called as proton motive force. So it is a ambit of energy that is present over there in the form of protons. So you have this force present over here. That force is able to rotate the so you have the ATP synthase. Now ATP synthase is also called as FO-F1 ATP synthase. So this component, which is the FO component of the ATP synthase, is rotated by the proton motive force, and in rotating it, two things happen. One, the proton is gushing in, so it allows the protons to come in through the ATP synthase, and second, it activates the ATP synthase, which is, so this is the F1 component of the ATP synthase, and that ATP synthase then converts ADP to ATP. So you're basically seeing that a force is being converted into a chemical energy. So you have energy conversion happening, and this ATP is then used as energy. When it is required, it is sent out from the mitochondria to the cytosol, to different organelles. And when it is and you can also have ATP being taken up by the mitochondria per se as well. But this is very well understood that the coupling of electron transfer and the ability of the oxygen to take up the electrons and phosphorylate ADP to ATP is what is called as oxidative phosphorylation, and that leads to formation of ATP. Today we know that if you have 1 mole of glucose, that can give rise to 32 to 34 molecules of ATP. If you have a fatty acid like palmitic acid present, then that can give rise to about 129 molecules of ATP. So you have since you have the NADH from glycolysis, NADH from beta oxidation, etc., that is formed, and those formed in the TCA cycle, giving its electrons to the electron transport system. We know that if NADH is giving the electron to the electron transport system, you get 2.5 molecules of ATP, while if you have FADH2 giving the electrons to the electron transport system, then you have 1.5 molecules of ATP formed. So that is something that makes mitochondria the powerhouse of the cell. Now let us look at the fact that mitochondria, apart from synthesis of ATP, also is a site for several different cellular metabolism. So, like for example, uh, NADH per se is not able to enter the inner membrane. So, however, you need the NADH from all the pathways that synthesize NADH. NADH can give the electrons to the electron transport system to give rise to ATP. So, effectively, NADH from the cytosol should come into the mitochondrial matrix. But it cannot come in directly because the inner membrane is impermeable. So, there are several shuttles that are present that enable the NADH to enter. So, as you can see over here, NADH and oxaloacetate gives rise to malate. Malate through a transporter is brought into the matrix. There it is converted back into oxaloacetate, and in the process you get mitochondrial NADH plus H plus. Now this NADH plus H plus will obviously give its electrons and protons to the electron transport system. This oxaloacetate is now going to go back in the form of aspartate. So this shuttle that is there is what is called as malate aspartate shuttle. The main thing is the buffering of the NADH, the making available to the mitochondria for oxidative phosphorylation the NADH, but in different forms. So glycerol 3-phosphate shuttle is also another way by which you can have such a system or such a transport of molecules that cannot directly enter through the inner membrane possible within the matrix. Uh, then you also have, two steps of urea cycle. So we know that urea cycle is responsible for converting the toxic ammonia that one gets from catabolism of amino acids. So, or the ammonia that is formed, the toxic ammonia that is formed, is converted to a non-toxic urea form, and that happens in such a way that 5 steps of the um, there are 5 steps, and of that 2 steps happen within the mitochondria, and this is especially seen in the liver. So there are two enzymes, that is the CPS1 and the OTC, ornithine transcarbamylase, and the carbamyl phosphate synthase. They are responsible for enabling formation of ornithine. Okay, and this ornithine then goes in further so that urea is formed. So therefore, this urea cycle also takes place in the mitochondria. Not the entire, but only 2 steps. But these 2 steps are important and crucial. Now, mitochondria, apart from synthesis of ATP, is a biosynthetic hub. So you can see that you have nucleotide synthesis, you have glucose and heme synthesis. So the mitochondria ensures that all these anabolic pathways happen within the mitochondria, and the precursors of many of these are present in the matrix itself. Similarly, you have fatty acid and cholesterol synthesis. You have amino acid synthesis also happening inside the mitochondria. And many of the membrane phospholipids like cardiolipin, phosphatidylethanolamine, etc. too are synthesized in the mitochondria. These phospholipids then move to the ER, where the other phospholipids are synthesized, and together they help in membrane biogenesis. There are very special functions that are associated with mitochondria. Now, mitochondria can enable a process which is called as mitochondrial fission. So, we all know that bacteria multiplies by what is called as binary fission. That is, an existing bacteria, by forming a central, furrow, will basically form two bacteria. So, uh, mitochondria also shows the same kind of phenomenon, which is called as mitochondrial fission. And as you can observe, you have the mitochondria dividing into two. So therefore, fission enables increasing the number of mitochondria inside a cell. Then you can have the other way around, that is, you can have two mitochondria fusing together to get to one. So the trigger for mitochondrial fusion could be under certain stress conditions, also aging, senescence, all of this may lead to either fission or fusion. Another important functionality is apoptosis, or what is called as programmed cell death. So, depending on a signal, a cell can, a mitochondria can give rise to reactive oxygen species, and also certain apoptotic factors which, when regulated, decide whether a cytochrome C has to be released from the mitochondria or not. If cytochrome C is released from the mitochondria, then it would activate a caspase pathway, which leads to the cell dying. So, apoptosis is also something that the mitochondria is involved in. Now, to another important feature of mitochondria is that the mitochondria has its own DNA. Okay, going back, because mitochondria shows shows a phenomenon which is very similar to bacterial fission, that is bacteria getting divided into two and mitochondria also getting divided into two. This is one of the factors that, or this is one of the functions of the mitochondria that is aligning it with a theory of, and the theory is called as the endosymbiotic theory, that suggests that mitochondria has arisen, or has arisen from a bacteria. So a larger bacteria may have taken up a smaller bacteria, and the smaller bacteria within the larger bacteria evolved to become a mitochondria. And that is why mitochondria is still able to carry out fission, because bacteria can carry out fission, mitochondria too can carry out fission. So that is how it is aligned with the fact that the ancestor of mitochondria could have been a bacteria. Another important facet is that the mitochondria has its own DNA. And looking at the human mitochondrial DNA, as you can see, it is about 16 kb. And this 16 kb DNA, you can see, has several structural genes which gives rise to important proteins of the electron transport system. So many of the proteins of the complex 1, some of the proteins of complex 3, of complex 4, and also the ATP synthase are synthesized by genes present in the mitochondrial DNA. So if there are mutations in these, then it can definitely affect ATP synthesis. And therefore, many diseases have been associated with mutations in the mitochondria. So mitochondrial DNA gives rise to its own genetics. That is something that has been prevalently studied. And in another interesting fact is that they have genes that also give rise to ribosomal RNA and tRNA. So you must understand that because they have ribosomal RNA, they are capable of forming ribosomes inside the matrix of the mitochondria. And so we therefore understand that mitochondria has its own ribosomes. And these ribosomes are of the 70S type. We all know that eukaryotes have ribosomes of the 80S, but mitochondria has ribosomes of 70S, which is again similar to bacteria, because bacteria also have the 70S type ribosomes. This is another evidence, or another factor that kind of gives a trust to the endosymbiotic theory, suggesting that it was originally a bacteria, because the ribosomes within the mitochondria are also the 70S type. So you very well therefore can understand that within the matrix you can have all of these proteins being synthesized by the 70S type ribosome, and also because it gives rise to tRNA, the tRNA are able to help in the protein synthesis. Of course, the mitochondrial DNA has the origin of replication, so therefore the DNA can multiply. So when you have the mitochondria going into fission, you would obviously have the DNA to have multiply into copies, okay, so that both the mitochondria have the mitochondrial DNA. So therefore, the number of copies of mitochondrial DNA within mitochondria can also vary. It has been, kind of suggested that the size of the mitochondrial DNA it may be different from the size of the bacterial DNA, because of the fact that there are several genes that may have been lost in the process of evolution. And now what is also understood is that the mitochondrial DNA, or the mitochondria, has to rely on several nuclear DNA for its functioning. It doesn't have all the proteins that is needed for functioning. So therefore, there are a lot of nuclear proteins. I mean, the nuclear proteins that are formed from nuclear DNA that have to be brought into the mitochondria. Here, this is just showing the fact that you can have the mitochondria, inherited from either parents. So, a recent study has shown that you can have both the father and the mother being responsible for passing on the mitochondria to the children. But majorly, what has been observed is that the inheritance is more maternal than paternal. So, there are more evidences of maternal inheritance for the mitochondrial DNA. So, let us make the conclusions. Mitochondria can be found within eukaryotes in many forms depending on what it is associated with and its function. The number of mitochondria too varies from cell to cell. It is a double membrane structure with both outer and inner membrane having structural and functional differences, enabling the mitochondria to carry out various functions. Mitochondria is an organelle that is a hub for both catabolic and anabolic pathways, with many of these integrated to enable oxidative phosphorylation, making mitochondria the powerhouse. Mitochondria has several features such as biogenesis, fusion, mitochondrial DNA and its associated genetics, role in apoptosis, etc. Mitochondria, like its folded inner membrane, has enfolded within itself several multicomplex systems to assist the entire cell and eventually the organism in several ways, especially supplying the currency of energy, ATP. Thank you.